For those who are not aware, she's actually part of the dynamic duo together with her husband Bill Miller, who are huge supporters of the APVRS. And I'd like to welcome both of them to Malaysia and thank them very much for making the trip from Chicago to share with us their experience today. Jennifer.
Thank you, Ken. I'd like to thank Ken and the organization committee for this kind invitation. Today I'm going to talk about the drug delivery system, particularly targeting the posterior segment, whether it's delivering anti-VEGF or other agents. I do have a U.S. patent and also international patent pending on the Microsphere thermoresponsive hydrogel drug delivery system, which I'll be talking about as part of my presentation. And I also do receive NIH research grant. So with the anti-VEGF being available for treatment, now we can administer monthly or even bi-monthly injections. And knowing that a lot of our patients will either maintain or improve their visual function. However, the burden of monthly or even bi-monthly injections on patients, physicians, or healthcare cannot be ignored. If you look at the number of intravitreal injections performed, in U.S., Medicare population in U.S., intravitreal injection was done about 4,500 injections in 2001, which increased to almost 2 million in 2012. And it has been projected this injection number will be close to 7 million in 2017. And this is just looking at US numbers. So this is actually a big clinical problem that we like to actually solve. We do know that in the eye is a very special place. We can actually deliver drug in multiple places, whether it's a topical, transcranial, or even intravitreally. But we do know that intravitreal injection is the most efficient, direct way to deliver to the posterior segment. But there are some complications and potential considerations you do look at, potential infections or detachment of the retina, cataract formations. But really the biggest challenge is the bolus amount of drug delivered to the eye. What do I mean by that? What it is, after injection, the eye is experiencing high concentration of the drug, which then with the short half-life, with the fast clearance, rapidly decrease the amount of the drug exposure to the tissue. So the eye is experiencing this high and low concentration, which can be either toxic or it can be ineffective. And what we really need is something that's sustained deliver over time in a smaller dose that can actually achieve a therapeutic goal. And we think that we can actually do this with the drug delivery system. Now, let's look at the current FDA approved drug delivery system, and I'm going to really focus more on what's coming in the pipeline. Really, if you look at the FDA approval system, there's only 3 drug delivery systems that can be currently used clinically. These are Retisert, Ozurdex, and Iluvien, and these are all solid implants that are delivering corticosteroid drugs to the eye, whether it can be delivered for several months to even over a year. And I'm not going to go into too much in details, but they do actually work really well by degrading the polymers that encapsulate these drugs over time. But what are the challenges? Why can we use those same system to deliver anti-VEGF? Well, one of the things that really make the anti-VEGF delivery system challenging is the large molecule size of the protein itself, as well as the delicate nature of the protein. So the interaction between polymers and protein actually can be quite tricky, as well as how much drug is going to be available and how long we can actually deliver because of this nature of the protein, and that actually poses a lot of challenges. So let's look at some of the drug delivery systems, and then some are going to be focused on anti-VEGF, some are actually on other drugs such as corticosteroid. The first technology I want to talk about is microneedles. These are micro-sized needles with the injector can deliver targeting the suprachoroidal space. Now, Clearside Biomedical is a company in U.S. that actually obtained FDA approval for clinical trials use of their device. Currently, they do phase 2 and phase 1/2 trial looking at the injecting translumin for the non-infection uveitis, looking at the Peachtree and a Dogwood trial. In their trial, what they noticed that there's definitely visual acuity improvement, reduction in retinal thickness. There's really no serious adverse events associated with the treatment, and really no steroid-related increase in IOP with these patients. There were some few minor adverse events were reported, such as decrease in vision, eye pain, or the cystoid macular edema. But overall, the system seems safe and effective. Using this technology, they're now moving into a retinal vein occlusion space. They're just sort of completed phase 2 study, Tanzania trial, where they looked at the injection of suprachoroidal TA versus in conjunction with anti-VEGF injections versus anti-VEGF injections alone. And what they found that the combination study actually requires 60% fewer injections of anti-VEGF, and with that, they're actually looking into phase 3 studies. Again, similarly, they're also looking at diabetic macular edema. We're looking at the suprachoroidal injection of TA alone with or in combination of anti-VEGF, and they're expected to have data at some time this year. The next system is a microcatheter. Again, microcatheter is targeting suprachoroidal space for the drug delivery system. So idea is that using microcannula, you insert through sclerotomy, and the lighted tip actually guide the surgeon to the targeted site. And you can then, once you reach the target site, can actually deliver the appropriate drugs or agents. Safety studies have shown that there's really no serious intraoperative or postoperative complication with this technology. Janssen Biotech has actually used technology to actually deliver the cells. This is really more for the gene therapy or cellular therapy rather than the drug therapy that we've been talking about, but it does have a really good outcome. Their phase 1/2 clinical study have shown that the use of this technology with the delivery of a human umbilical tissue-derived cells actually really improved the dry AMD patients. The patients received 50 microliters of the cells in a single injections, and the eyes that received the treatment improved 4 to 5 letters were compared to untreated eyes, and there was really about a 15% rate of retinal detachment, but really no other negative side effects. So again, this is not for the monthly injection of anti-VEGF, but perhaps for gene therapy or the other cellular-based therapy. Encapsulated cell technology uses a combination of the material as well as the cells. The system consists of cells that have genetically produced the particular drug they're actually encapsulating this semi-permeable hollow fiber membrane. So these fiber membrane will actually protect these cells from the body as well as allow the drug or the agent to diffuse out of the implants. And these implants are placed in vitreous and can be retrieved, and they've shown that they can release up to about 18 months. New York Tech is a company in the U.S. that actually produces and studies the system, and NT501 is their hallmark study. Looking at a phase 2 study, they've shown that there's increased retinal thickness and stable vision in the treatment of GA associated with the dry AMD and RP patients, and some patients actually had positive impact after 50 months after the post-implantation. They're also looking at the phase 2 study of MACTAL as well, and so this is actually something they're continuing. Now, with this positive result, they now move into look at how to inhibit VEGF. So they actually remodified their system a little bit more to allow a lot more cells to be encapsulated within the technology. And their phase 1 study, the safety study looked really good with some patients that showing this clinical efficacy with the system. They began a phase 2 study with a higher dose, but unfortunately they actually discontinue it in 2016 due to the large number of patients requiring the medical injections. So unfortunately they are not going to be pursuing this technology for the anti-VEGF, but this is something we can perhaps use for other agents. Replenished micropump is an electromechanical system that can actually deliver either continuous or bolus amount of drug, whether to the anterior segment or posterior segment. This is a reservoir that sits outside of the eye and that is connected to the anterior or posterior segment by the cannula. And what's really interesting about the system is that you can actually program it. You can program it to deliver once a day or continuously throughout the day, and you can actually refill the reservoir using small gauge needles. Mark Humayan and colleagues are the ones that are actually developing and testing these systems, and their recent study actually demonstrated safety and surgical feasibility of the pump in patients with DME. In fact, their study has shown that it's safe to implant and for a period of about 90 days, and the implant was able to deliver anti-VEGF dose within the 20% of the targeted amount in 7 out of 11 patients. So they will continue to look at this technology in the space. For the next couple minutes, I wanted to actually talk about some of the things that we're doing in my laboratory, which is using the Microsphere hydrogel delivery system. So I have a biodegradable microspheres that can actually encapsulate anti-VEGF or any other agents, and that's actually embedded into biodegradable thermoresponsive hydrogel. And hydrogel will confine the microsphere to delivery sites as well as provide another layer to prolong the delivery. Now, the hydrogel is interesting that it's a thermoresponsive. So at room temperature, it's a liquid-like, so you can actually inject using small gauge needles, and it will become a solid gel at body temperature. And we're looking, we can inject it intravitreally or subconj or even topically. We have recently shown that our system can actually have initial bursts of about 13 micrograms of aflibercept, then continues to release about 0.065 microgram per day for over 200 days. That's over a 6 months period of time. Not only that we can release for 6 months, but the release sample is bioactive. In fact, we have demonstrated that they can actually reduce the CMV lesion size in rodent models. And this similar treatment efficacy as the bolus injection of the aflibercept. And we have also demonstrated they can actually also work for ranibizumab as well. We're now actually looking at the non-human primate mice to see if we can actually demonstrate further the efficacy of our system. We know that we can deliver over 6 months in the monkey eyes with the constant level about 0.37 micrograms per day of aflibercept. We also looked at the safety. We inject these eyes and follow biweekly for 6 months, looking at the OCTs, ERGs, and FAs and clinical exams. And here's an example of OCT compared to control eyes, where there's no difference between the treated or untreated eyes, and when OCT or ERGs or FAs. And here's a histology of the at 6 months. And this particular animal, we inject a non-degraded hydrogel so that you can actually see in our histology, again, there's a very minimal inflammations or changes to the eye. So finally, I'm going to like to summarize. There's a variety of approaches to improving drug delivery system, and I'm only talked about very few. There are many more technologies and techniques out there that are trying to address this problem. Intravitreal injections and the use of the solid implants are the only thing we have right now, but I do believe that the advances will continue to occur with either new drugs or the new techniques to deliver these drugs